The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. In today's lesson, we will talk about patterns of genome variation, focusing on mutations. Mutations arise from replication errors and base modifications. They are due to the fact that replication is imperfect. Sometimes, DNA polymerase occasionally will insert incorrect nucleotides, and then these misincorporated nucleotides will persist after proofreading. Errors due to mispairing predominantly leads to point mutations. Now, there are two types of mutations based on origins. They're either spontaneous mutations. These happen naturally and randomly. They usually link to normal biological or chemical process in organism, but altering structure of nitrogenous bases. These occur during enzymatic processes of DNA replication. There's always some rate of spontaneous mutation in your genome, as opposed to induced mutations. Now, these happen because of the mutagens. The most common mutations include base substitutions. We could also call them SNPs, or single nucleotide polymorphisms. These are binary mutations with low average nuclear rate of the order of 10⁻⁸ per base pair per generation. Among these, there is a bias towards transitions over transversions and a tenfold elevated rate of CpG dinucleotides. Base substitutions which lead to a change of amino acid are termed non-synonymous or missense mutations. Most base substitutions and other variants are selectively neutral. These silent site or synonymous substitutions are also often assumed to be selectively neutral. There are many types of classes and scales of mutations. Mutations result in a variant form that may be transmitted to subsequent generations caused by alteration of single base pair in DNA are deletions, insertions, or rearrangement of larger sections of genes on chromosomes. While single nucleotide polymorphisms are widely distributed and frequent, other cases like microsatellites and non-GC rich minisatellites that are also very widespread. Base substitutions in genes can adversely affect gene function through amino acid substitution, including inclusion of stop codons and regulatory or splicing changes. Mutations outside genes can have functional consequences in less well understood ways. Base substitution data comes from individual genome comparisons. Initially, SNPs were discovered from analyzing a few loci in several individuals, and as a result, many of widely used SNPs have diverse and poorly documented origins. Now, whole genome resequencing studies are revealing relatively unbiased pictures of numbers and distributions of variants between genomes. For example, 1000 Genome Project uses a combination of low coverage, as low as 4x, genome sequencing with genotyping and deep targeted sequencing of all coding sequence in 2,500 individuals from major regions in the world. Mutations are identified by aligning sequences or aligning sequence reads to the reference genome. Germline mutations are inherited from generation to generation, while somatic mutations arise in tissues during individual lifetime. Some of these somatic mutations can lead to cancer. Somatic mutation is used in tumor samples. For example, DNA sequence read from a tumor sample is aligned to reference genome shown here in gray. Single nucleotide differences between reads and reference genomes indicate germline single nucleotide variants, or SNV, in green circles, somatic SNVs red circles, or sequencing errors black diamonds. The rate of base substitution can be estimated either directly or indirectly. The mutation rate in nuclear DNA is often so low that until recently it was impossible to measure directly in pedigrees, and indirect approaches were therefore used. For example, there was comparison of non-functional DNA sequences between species whose divergence times were known and give an estimate of mutation rates. For example, an estimate was given from studying 2 million base pairs of non-contiguous chimpanzee sequence compared to the human orthologs. In CpG dinucleotides, where a C nucleotide is followed by a G, mutation rate is elevated by more than tenfold. Most of these CpG dinucleotides are methylated at the 5 carbon of cytosine on both strands. Consequently, the rate of base substitution will not be uniform throughout the genome. It varies significantly genome-wide. In principle, this could reflect variation in mutation rates because, for example, of variable CG contact or variable efficiency of DNA mismatch repair, for example. However, as it's discussed before in lectures 5 and 6, the sequence diversity of a locus is influenced not only by its mutation rate, but also by natural selection and population history. Another place where mutation rates are observed at higher rates are microsatellite loci. Microsatellites are 2 to 9 nucleotide tandem repeats in DNA sequences. The number of repeats is variable in population of DNA and within the alleles of an individual. Microsatellites usually form due to slippage and misalignment. Defects in mismatch repair, such as those in some human cancers, can lead to elevated mutation rates in microsatellites as well. Here, mutations are shown in single-step increases or decreases. Mutations involving greater repeat numbers occur, but are about tenfold rarer and may involve other mechanisms, not necessarily slippage. How do we identify microsatellite loci? Despite their utility, microsatellites are particularly difficult to genotype. The repetitive sequence responsible for their high mutability also results in frequent alignment errors that can complicate biased downstream analyses. Programs like Hipster were specifically developed to deal with these errors in the hopes of obtaining more robust microsatellite genotypes. Do not confuse microsatellites with mini-satellites. A mini-satellite is a tract of repetitive DNA in which certain DNA motifs, ranging in length from 10 to 6 base pairs, are typically repeated 5 to 50 times. Here is an example of 2 mini-satellites. One, an autosomal mini-satellite B6.7, an interspersed pattern. Each different colored circle represents a sequence variant with a 34 base pair repeat unit. On the other hand, we see the Y-chromosomal mini-satellite MSY1, which shows block-like patterns. Each different colored circle represents a sequence variant on a 25 base pair repeat unit. Many satellites occur at more than 1,000 locations in the human genome, and they are notable for their high mutation rate and high diversity in population. These have been associated with chromosome fragile sites and are proximal to a number of recurrent translocation breakpoints. Examples of mini-satellites include telomere arrays which are at the termini of chromosomes, and most GC-rich minisatellites are concentrated towards chromosome ends. All chromosomes carry alpha satellite sequences in their centromeric regions, although there are other satellites on specific chromosomes at other positions. Transposable elements are other type of variation. Specifically, in a human genome, LU elements are concentrated in GC-rich regions. These are shown with light G bands, and L1 elements are concentrated in AT-rich regions, which are shown in dark G bands. Recent insertions of both elements are concentrated in the latter regions. Structural variation is variation of structure of DNA regions affecting DNA sequence length and/or orientation. It generally occurs as deletions, insertions, copy number gains, inversions, and transposable elements. How do we identify structural variation? There are four main methods for detecting CNVs in next-generation sequencing data. First, read pair compared the average insert size between the actual sequenced read pairs with the expected size based on the reference genome. Second, split read reads from pair-end sequencing where only one read of the pair has a reliable mapping and the other one either completely or partially fails to map to the genome. Read depth. This one is based on the hypothesis that there is a correlation between depth of coverage of a genomic region and the copy number of the region. Finally, the assembly-based method, which first generates a contig/scaffold that are then compared with the reference genome to discover structural variation. Here's an example of genome-wide detection of CNV. The lower plot shows an example of genome-wide CNV detection using oligoarray comparative genomic hybridization. Red and blue arrows represent copy number gains and losses, respectively. Different classes of polymorphisms have different mutation rates. The mutation rate in nuclear DNA is often so low that until recently it was impossible to measure it directly in pedigrees, and therefore indirect approaches were used. Then comparisons of non-functional DNA sequences between species whose divergent times were known could give estimates of mutation rates. Today, we use next-generation sequencing for comparison of sequences between individuals and populations. Here are some review questions for you. Describe mechanisms leading to mutations. What are the base substitutions and what are their possible effects? What is the overall rate of base substitutions in the genome? What are the microsatellites and how do they form? Where can you find many satellites in the genome? Describe types of structural variants. What are the mutation rates for the fastest and slowest changing mutations described in this section? This concludes today's lesson, Patterns of Genome Variation Mutations. Next, we will be studying the topic of patterns of genome variation recombination.